Since yesterday there was some lag in the internet, so what I will do today, I will switch off my video and then we can start the lecture. Okay. So let's start for today's lecture. What we have been seeing in last two lectures, especially from lecture number 13, that we are talking about a tail, right? That is transcription activator like effectors. So basically, in first lecture, that is lecture number 13, what we discussed in first lecture, let's see it once again. So in first lecture, I told you a brief summary about the relationship between avirulence gene, AVR gene, and what is the relationship between plant immunity. So that means how this avirulent genes affect plant and how it enters into plant for making it susceptible. But if plant has the resistance gene, then this resistance gene is able to detect its effectors, that is avirulence effector, and this has to be cognate. Cognate means the same kind of resistance gene should be there to detect this effector gene. So this was the first relationship we actually discussed in our first lecture, where we also saw about pattern triggered immunity as well as effector triggered immunity. These two things we have seen in the first lecture, and that was the complete picture where we discussed AVR, binding with resistance, and then how these two compete. If one wins, that means you have either susceptible plant or you have resistant plant against the effector genes. That was the first thing. Then in the same lecture, we also saw that how subsequently this AVR genes were isolated and sequencing was performed. And the prominent feature from sequencing was detected that they always found this special repeats kind of sequences. So for this important repeats, they were able to find, and what they conducted after that, that these repeats were deleted. For example, certain portions were deleted or mutated, and this kind of studies then they started to perform. And from this kind of study, they were able to conclude one thing, that the number of repeats, then the order of repeats, and the length of repeats, all these things are important in creating your hypersensitive response. That is, whether the plant is susceptible or the plant gives your local cell death, it always depends on this number of repeats. If you delete, then you do not have hypersensitive response, and if you have proper repeats by swapping experiments, then you have this response. So the number of repeats, order of repeats, and the length of repeats are important for the this response. And from there, it was concluded also that the sequence specificity is always directed from the repeated sequences, or these sequences shown here in the red mark here. So these repeats were found in our lecture number first, in case of tail, when we started story of the our tail protein. So that was the two prominent experiment I have shown in the first lecture, where after this sequencing also they deleted and then they concluded up to this. So that was the whole part of first lecture. Now in second lecture, this is the part of second lecture. That means after concluding that these repeats are important, what are the other features of your avirulent genes? So other features or other structural features of avirulent genes were presence of your nuclear localization signal and presence of your acidic transcriptional activation domain. And for them, they concluded this is very much a part of eukaryotic like motifs, and these are important also for your hypersensitive response, because if you delete this mutant or delete this nuclear localization signal, or if you delete some of the portions of hydrophobic domains in the acidic transcriptional activation domain, then this response is deleted. And this we have seen in the last lecture, where the importance of NLS2 and NLS3 was much greater than compared to your NLS1. So this was the part of our last lecture. And then after that, we also saw in the end of the second lecture that these AVR genes actually, when it enters into the plant cell, it upregulates some kind of proteins. And basically these proteins were called as UPA gene, or UPA gene is upregulated. I have shown here as a mark, and these proteins were upregulated. And most of these proteins were, you know, found as UPA7 and UPA 20. Especially we have seen these two. And what has been found that UPA7 involves in the expression of alpha expansin. I will write it here, alpha expansin, and these are also important. UVA gene is also important for creatine auxin kind of cells. And these important proteins are involved in the enlargement of your cell. So that means especially mesophil. And you know the relation between mesophil and your infection, because your infection starts with the opening of these kind of natural opening of the pores. For example, one mesophil is one of them. And once they started finding out these direct targets, they were able to show that all these genes which are upregulated, especially UPA7, UPA20, they all contain a specific promoter sequence that was named as UPA box. So they were named as UPA box. So these were the conserved promoter sequence. So when they found this conserved promoter sequences, they also did experiments where they showed that your AVR proteins are able to bind here directly on the conserved UPA box. That means UPA box are the direct targets of your AVR proteins. So once they did this kind of experiments, they proved it with the help of footprinting assays. Then they proved this as well as from the chip assay and also from the help of EMSA. So all these experiments they did for showing the direct binding of your protein with your promoter sequences, conserved sequences. Now, as soon as they found that all these avirulent genes are binding to UPA boxes, so close observation, they found that UPA box length, for example, is always corresponding to its repeated sequences. For example, UPA this is AVR BS3 containing 17.5 repeats. So exactly your UPA box would contain, the length sequence would be around 18 base pair. So these kind of observation they were able to find, and it was also important from the above experiment, especially in lecture 1 we have seen, if you know interchange the repeats. For example, this is 1 2 3, if you change the third repeat to the first position and first repeat to the fourth position, and if you interchange within the repeat, it is not possible by the repeats to bind the same sequences then. So the order, especially the order, number and all this length were important for the repeats. And then the close observation that this always matches the length of UPA box target, that means, and the repeats within the AVR aviral genes repeats is always equal or matching. And always they also found that UPA box is one base pair longer than your repeats. If this is 17.5, then you always have a 0 position here, and this always starts with T. So there is a base pair here as a T. So this much we have seen in first 2-3 lectures, and now we will see further. So this is your UPA box as I showed in the last lecture, and this has been shown. Um, these are, so that means in last lecture we showed that the structural domains of your AVR. What are the important structural domains? That means repeats are important, then you have NLS is important, then you have AD domain is important, and even after that you have left and right some kind of inverted repeats, right? So these repeats of 62 base pair were also important. Then you have at the N terminal region, you have translocon signal, so that transfers your whole protein into the cell. So all these structural domains we have seen so far. Now going further, moving further, how this UPA20 was shown as a direct direct partner of or direct target of AVR gene. So that was also shown. Number of experiments done by UPA RT-PCR was shown. UPA7 was a direct partner, then direct target. UPA20 was a direct target, and mutating these would not lead into any hypersensitive response, as we have seen in last lecture. Then they were able to, you know, check which is the minimal region for the promoter sequences. So all this we have seen in our last lecture. So the close observation what showed them that there is always a corresponding repeats as well as sequences. That means UPA box binds with your repeated sequences. And even further, when they noticed in detail, they always found that the 12th and 13th amino acid within the repeat is important in binding to one base pair, sorry, one base pair here as shown here. So basically what they were able to deduce manually, that HD always binds to your base pair A, HG always binds to T, NS also binds to A, C or G or T, that we have shown in other experiments. NG is always binding to T. So this kind of experiments they were able to show with the help of Gus reporter system, where they mutated their target sequences and they were able to find out what is the important base pair that is binding to particular amino acid. So there was some kind of, you know, pattern. For example, NN always bind to NG. So with this pattern, especially with the preliminary work on AVS-BS3 and UPA box, they were able to, you know, deduce that what are the next promoter regions. For example, if they know the sequence or a repeated sequence of one of the pathogen effectors, for example in this case HAX2 repeats are also already known, for example NNIG, and if this is known, and then based on these sequences they were able to find what are the promoter region for HAX2 box, and similarly for HA3 box and HAX4 box promoter region were found based on the homology work and based on the sequences. So then they performed an assay where reporter systems were created, where they tagged their promoter region with your Gus reporter system, and with the help of color, basically blue color, they were able to find that only cognate pairs, for example only effector gene having HAX2 will only induce your HAX2 promoter sequences. It will not induce HAX3 box, as you can see here. There is no induction here. So only when HAX3 is in effector is infected, then you have upregulation of genes which are based on the HAX promoter boxes. So these kind of experiments prove that it is a cognate pair that is working, and what are the direct targets of HAX box. So from this, they even went further and were able to find what are the important base pairs that is prominently binding to di residues. That means these are di residues at 12th and 13th position. So what are the different residues? So basically Ng always binds to T. It doesn't allow any other base pair to come along, whereas Nd binds to C always, and no other base pair is allowed. Similarly, Ni binds to A. Whereas when you see ns, that it always binds to either a or all other sequences, or all other base pairs. So here, and similarly there are other residues, for example nn and for example here nn, nk and nh, they all have some kind of affinity towards guanine residues. So with this they were able to find the basically code. So code means how these repeats are able to find targets of or particular DNA. Now if you have certain kind of this protein where you have tail proteins, right, transcription activator like proteins or effectors, so you can also produce one artificial tail. So instead of prominent tail, you can produce any artificial tail, and basically you can target any of the gene which is required. And here you don't have to depend on any other residue, because the code is very much simple. For example ni exactly at 12th and 13th position, then ng 12th and 13th position will recognize these pairs. And there were also experiments done whether other residues apart from repeats are helping in your binding assay. So all other residues were not very much specific to base binding. They will so I will come to it. So basically only the repeats were binding to the targets specifically. So site specific targets were only created by your dye residues which were present at 12th and 13th positions. So based on this, one can create your artificial effectors with novel targets. And because of this simplicity in nature, this molecule was considered as a molecule of the month in 2012 after its structures were created. So as soon as as soon as people knew about the UPA box relationship with its repeats, they were able to deduce its code, that how it will bind to its targets. So these were also known. And soon after this work on UPA boxes, people started working on the crystal structure, and the structure looks something like this. So what you see here is your a novel way of DNA binding domain that has been deduced with the help of crystal structure. And basically two independent groups, Moscow and Walk, both in 2012 I think, they were able to deduce the whole structure on the basis of this. So what you see here is a remarkable staircase like format. So what you see here from the front view, and from the top view, what you have here is the helical region, then the loop is inserted inside, and then you have helical region. So the structure of tail was deduced at the helix loop helix structure, right? So especially uh, when you see closely here, so when you see closely the 12th and 13th residue of one of the repeats here, and when you see the structure, so especially the 12th and 13th residues are the important ones which are placed on your loop position. And this loop is inserted inside your DNA sequences. For example, this is DNA helical region from top position, and these are connecting into the bases. So here all this connection are taking place into the major grooves, as you can see here, and the only the loop position is able to have a contact with your base pair. And if you notice here, from 12th and 13th position, it is only the 13th position which is facing towards your base pairs. So the 12th position amino acid residue folds back to its own structure, especially stabilizing the loop. So its role in is there in stabilizing the loop structure. It doesn't have any role in contacting the base pair. So basically it is only the 13th residue which is taking your, which is important in stabilizing or site specificity, I would write site specific contacts. So this is done by only the 13th residue, and the 12th residue folds back on its own structure forming a loop, and it is always stabilizes the loop. So based on this, they were able to deduce what are the residues present in whichever repeats. For example, here HD is shown, H folds back on 12th and D is making contact with C. Then here HG, then this G is making contact with T, and so far and so on. So these are the prominent structure which they were able to deduce from the crystal structure. So as I mentioned already in my previous lectures also, that the molecular details is always possible only after having crystal structure, because the detail, even biochemical studies are very much close in alignment with the structure what we got here. So but even only after the crystal structures were obtained, it was found that it is the role of 13th base pair which is important for making a site specific contact, but the di residues are always contacted together. For example, NN, HD, Ni, all these residues are important. So this is the whole story of your transcription activator like effectors, but there are few things which I have not mentioned so far. One thing is important here, that we always see a T residue at the 5 prime end, and why we have only T residues in the end. This is because if you see, this is 23.5 repeats present in this structure. Then before this structure, you also see some kind of 0 and 1. Okay, so these are not complete structure. These are half and half structure. That is why it is reported. So for example, this is 0 repeat, and together 0 and 1 repeat, or 0 and minus 1 repeat, forms a structure similar to 1 or 2. That means helix, loop, helix kind of structure. So these together forms helix, loop, helix kind of structure, and that means 0 and minus 1 repeats are together forming a total structure that is correspondence to your 1. And both together, they are actually binding to T residue or T base pair. So here, because of the presence of tryptophan at position 232 in this case, and this position 232 or tryptophan is placed especially at the loop position, also in the -1 repeat, and because of that you have the T residues. And people later found out what is the reason, but I have not seen why exactly this T is present, but it is always present in the this repeat forms. And then there are other details also. For example, the last residue is always half residue. So I have also mentioned this in the first lecture also, because the conserved sequences were only present until half sequences. So the last residue is always half. So that is why it is 23.5 or 17.5 or 11.5, something like this. Now there are certain residues which are known as 127 and 1149. So these residues until here, they are important, and I will come to it in subsequent slides. So with this, now you have certain protein altogether that has a very simple code. And now, like you have had the fork domains. For example, this is an example of your zinc finger protein, which was linked with your fork cleavage enzymes. And this, with the binding of cleavage enzyme, it becomes zinc finger nucleases, right? So now you can imagine your tail to be acting as a similar, where you are using the complete tail proteins and you are binding it with your fork cleavage enzymes, so that you can target these novel sequences. And these sequences, whatever sequences are present here, you can make your own kind of repeats. And since because this making this kind of protein was simple, and that is why this whole molecule was named as a molecule of the month in 2012. So by the time talents were, popularized and people started working, already CRISPR were all on the go. That people had discovered the role of CRISPR and how it was much easier even compared to talent to go and work in the laboratory kind of setup. So we will see CRISPR after this, but let us finish our talent. So these are the central repeats which are binding here. So the first talent, that means uh, nucleases binding to fork domains here, was constructed by the natural occurring talent. That means there was no deletion or mutants created. So that means the whole protein that was present in your talent was created. This is N-terminus sequence, this is C-terminus sequences, as you can see, and then two different targets were created, joined together, and expecting that it will bind to different targets and make a heterodimer kind of situation. So the first talon was created with the help of total proteins. So what you have here is your total N-terminal sequence. Then what you have here is C-terminal sequence. So if you imagine that 288 is really big protein. So you can imagine this has very big protein, 295 amino acid in the C-terminals, and in the spacer 16 to 31 base pair. In this region, 16 to 13 one base pair, and then this was created with the help of so the first talent was created by Lee et al and published in 2011. So based on based on beta galactoside assays they had performed. Now further down, lot of experiments were done where they wanted to check with the help of N-terminus deletion or C-terminus deletion, and even the smaller spacers. So the idea in engineering is to develop a very small molecule. See, if you have a very big molecule like 288 amino acid in the N-terminus, C-terminus you have a bulky amino acid. So all this is very difficult to be transfected in some kind of organism or cells. So therefore, the always preference is to construct a smaller proteins with a easily transfectable thing, and then easily mutating the region what is required for the editing kind of situation. So they deleted lot of sequences from the N terminal, and the last things they were able to mutate was up to 153 amino acid and up to 136. Below this, the if they got below 127, the whole thing collapsed. That means it didn't work anymore. And then from the C terminus, it was seen that up to 18 amino acid was important. So basically, after the first talent, there was another important what you say talents were created, where this was most popular, where 136 in the N terminus, 63 at the C terminus, and 12 to 20 spacers, amino acid in the spacer regions. So that was like you will find lot of publication with this kind of talent used in their system. And after this work, after Miller's work in 2011, there were other kind of residues that were even smaller compared to Miller experiments. So for example, in 2012, they had even better system, and in this case 153 and 17, so, and very small spacers, if you can see, up to 12. So those things were created. Now if you see that all effective talents had at least 127 amino acid preceding the central repeats unit. So if you see this picture, I had mentioned about this domain. So all these repeats were important, but before the repeats, until these sequences were also important, and people were not sure why these sequences were important. So if you delete anything till here, it will not work. So you will have to construct N terminus which should have up to 127 amino acid in your talent sequence. So it was found that all this N-terminus sequences, they also construct have something like the helical structure, helical loop structure, and these were numbered as -2 and -3. Although till 0 and 1 together they are specific for base contacting to the T residues, but the -2 and -3 was found to contact DNA. That means it is binding to the DNA, as you can see here, but it is not site specific binding. That means it is not able to contact any of the base pair, but for it is important for specific uh, uh, just for binding of your DNA. That means these structures are also important for the, apart from repeat sequences, -1, -2 and -3 sequences are important, and -1 is for T residue, but these are in general prominent binding to the DNA. So they were able to figure out with the structural residues. So this together closes the chapter of talent. So if you have any questions, you can ask. Otherwise, I will move to important topic that I will describe in 4 5 slides. Any questions? Please go ahead.
Hello, ma'am. Good afternoon.
Good afternoon.
Ma'am, what is the significance of the spacer region and where it is present?
Spacer reason. Okay. So you have, Amit, you have studied about zinc finger domains, right? So there I have described the spacer region was found to be around 6 base pair, minimum 6 base pair, that works pretty good with your zinc finger proteins, right? So if you have one molecule, so I will just write it here. Are you able to recollect that lecture? So where I
Yes,
described,
ma'am.
yeah, where I described this is your left zinc finger nuclease, which is bound to your fork enzyme. Then you have right uh, zinc finger domain, and when they come together they need at least 6 spacer region or 6 base pair region. That is important for them to find a dimer and to cut exactly at the prominent position to give you four base pair overhang. So that I showed in the zinc finger nucleases, and in fact if you read in that paper, they actually standardize the technique from starting from 4 base pair until I think up to 50 base pair. So in between they were able to find that the flexible linkal region is flexible to also make a cut at 6 base pair. And then it also cuts when 8 base pair spaces were created. Also it will cut at 16. So efficiency was there for all up to 48 base pair was the maximum they were able to find. So similarly here, if we are talking about the scaffold optimization, that means they are doing an experiment where they are checking which are the prominent spacer region, that means in between region, where you have to bind with your FOC1 enzymes, for example here, and how much is the minimum residues that are required for FOC1 to come and make a dimer kind of situation, so that both both molecule target to its prominent target site, and these FOC enzymes are also able to cut at the proper sequences, and you get an overhang so that you get further downstream processes happening, right? So that is the region for spacer. And in fact if you have longer spacer, that is much more what you say, you have to deduce that where exactly within the spacer region the FOC1 will bind exactly. And if you have lesser spacer region, that means you have to create your protein in a very compact way. You can create your protein in a much smaller fashion. So therefore if you see the 12 spacers was used in the later sequences, for example 153 at the N terminus, 12 at the spacer region, with the 17 at the C terminus residue. So similarly here the smallest is 136 amino acid, then 18 and 13 to 16 spacer regions. So these were the prominent exercise they would have done. And I have not gone into all the all the articles like Christian. I've gone to Miller, but not all articles I have gone to and seen their exercise. But they did this kind of exercise to know which of the talents is the best in their system. So if they use in the east system, this is best. If they use in the cell lines, this probably work best for them. And in the HEC 293, this probably works best. So based on their system as well as the kind of repeats they are making, different kinds of talents is there in the market, and you can use it for your study.
Yes, ma'am. Clear.
Okay, Sam.
Yes, ma'am. Ma'am, I had this question. We mentioned about NTS and the CTS sequences being reduced, amino acid sequence in the sense. But with respect to the repeated domains, is there any chance that we can possibly tinker that? I mean, reduce the number, or is it fixed that you have to have these many particular repeated domains in order to for the talent to function as such?
Okay, that is a very good question. So when you talk about the nature, see, nature is really fantastic because you get a lot of what you say, lot of molecules from the nature. For example, we got our fork enzyme from nature. We got our zinc proteins from the nature. And if you talk about the talent, so if you are talking about then in respect to your pathogenicity and respect to your this resistance, you cannot play with that. That means if xanthomonas is affecting, if you mutate in the xanthomonas sequences, this repeats, it will not be able to give their avirulence gene into the plant system. But here what we are using, we are taking that transcription factor, we are reproducing in E. coli in high number, purified form, and we are using and mutating it with our own benefit. That means we can change the repeat, we can change entire sequences according to our own target prediction. So that means if you, so there is lot of application for your talent. For example, you have this protein of interest. That means if I can create a repeated sequences, for example, you can say there are 10 repeats in this. So 1 to 10. That means it will bind to any sequences of my choice that I want to bind. For example, I'm writing it T, A, G, C, A, whatever, C, G, T, something like that. So I want to bind to this sequence. So what I want to do, I don't know, but what right now what I have in mind, that I want to bind to these sequences with one protein. So that means I can create one artificial protein with this specific repeats inside this tail, and then this will bind here. And now I want to check whether upstream of these sequences, whether some of the genes are behaving as a upregulated genes or downregulated genes. So that means if I want to tag any of the protein here, for example, activator, so I want to check one of the protein of my choice. For example, I am working on gene X or gene A or gene Y, and I want to check whether these genes are important in activating some kind of gene here which is upregulated. So what I can do, I can tag this according within this repeat sequences, and then check whether my gene of interest is behaving as an activator, behaving as a repressor, or behaving as a nuclease, if you can say so, or if it is behaving as a methylase or any other thing. So there are a lot of application based on this. So what we are doing right now is, we understood a lot about effector molecules in detail, from biochemical assays, from all different assays up till crystal structuralization. That means we know exactly that how this molecule behaves. And now is the time that once I know that this molecule behave like this, whether I can use this molecule of our own choice in the system. That means in vitro or in vivo experiments. So these are the prominent applications for this protein. So what I'm showing you is only for the editing, but there are a lot of things you can play with your zinc finger proteins or with your talon proteins, right? Tail effectors. So yes, you can change it according to your wish if you want to conduct any experiment of your choice.
Okay, ma'am. But ma'am, I have another question. So
Yes.
when comparing this with zinc fingers, wouldn't it be preferable to use zinc fingers because of the less, protein size, and as well the fact that each finger can actually take care of at least three amino acids?
Yes, in practical, if you say the zinc finger is whether it is, complex, but if you see compared to zinc and talon, I can tell you already that there are some pros and cons of using zinc or talon figure. So zinc pro is it is less in number or maybe small, I would say. Oh, sorry. So zinc finger proteins are small. This is large protein, right? But when you go for the code, right? When you have to construct your artificial protein, then constructing your tail is much easier than your zinc finger proteins, because for zinc finger proteins you have to go in sync with -1 3 and sometimes 5 residue or 6 residue. So these residues which are very much prominent, and sometimes although we say that it is modular, but sometimes it can happen that if you have at position 3 one residue, then in the next subsequent zinc finger repeat, you cannot have certain amino acid at that particular position. So because of this complex nature of zinc finger nucleases, this talon was much preferable than compared to zinc finger proteins. Although the cons is it is really of big size, and if you are able to sufficiently, people have used it in actually a number of cases, so they were able to send it towards into the organism or into the cell lines. So and that is why it has been also awarded as a molecule of the month. The only thing that by the time people started, deliberately lot of people started using tail. So 2012 was the time when the CRISPR was also in the hype. So 2009 I think was the time when CRISPR was first announced in the subject paper, and after that it started really booming, like everybody started using it. So that was the time, and of course compared to zinc finger, our tail is much better.
Okay, ma'am. Thank you, ma'am.
Okay. So I have few more things to actually finish. I will come to it immediately. And if you have any questions, please write it down. I will take it after finishing the lecture. Okay. So now we are talking about our cleavage domain of FOC1 enzyme. This is the cleavage domain, and as we said that it requires 6 spacers. But I was expecting this question, and many of you have also asked in terms of this question that uh, what are the prominent interface of FOC1 enzyme? And there was one question that I think 2 or 3 times people you guys have asked this question, that why after deleting D483 or also R487, which are the prominent interface in circular kind of DNA, why you do not get linear DNA, and why from complex uh, concatenated DNA, no, not the super coiled closed structure DNA, why you get a closed circular structure, right? So there I mentioned that in the interface of FOC1 domain, you can remember this slide where I've talked about the structure of interface.
Yes, ma'am.
Okay. So there I had also showed that apart from these prominent aspartic acid and arginine, there are other amino acid which are also taking place for the making a dimer kind of situation. That means your dimer interface is not only dependent on these two amino acid. There are other amino acid which are taking place. That means if you mutate these amino acid residue, there are still dimer which are taking place. That means still dimer is able to form. Now the question is, if the dimer is able to form, then how you are expecting your left zinc finger protein to, only get stable heterodimer with the right zinc finger protein. It can happen that you are able to form your one protein with the cleavage domain FOC1 with same protein with the comment. That means you are able to form homodimers because the other interfaces are also present. That means you can form the similar homodimer, and also the right zinc finger proteins is able to form the homodimers, and also they are placed with the fork dimer are also present, so they are also able to cleave. So this kind of question nobody asked, right? Although you asked it, but yeah, not specific to this question. So now we are touching on this prominent subject. So people saw that when you transfect this left and right zinc finger proteins or talon, then they were able to get homodimers as well as heterodimers, and the efficiency of getting specific heterodimer, because what you want in your experiment, that if you want to target one right right basis of this many sequences, then this protein should only bind here, and this should not go to any other sites in the complete human genome and not give any off target effects. So you do not want any off target effects, and for that reason you have form this heterodimer kind of situation. Left sequences, other target should bind into the right sequences, and the both dimers should form in that specific form where you will cleave only this kind of DNA and not to other kind of DNA. So they were facing this problem, and thus the efficiency of zinc finger dimers were actually improved after knowing the intricacy about the this dimer interfaces. So what they wanted to create after that, that only the left will bind here and the right will bind exactly at here, and the cleavage will happen only when it will form the heterodimer, and not, there is no cleavage. If you can see here, there is no cleavage, and no cleavage when homodimers are formed. So how kind of molecule, when you kind of molecule is called obligatory cleavage. So that means you want to cleave only when this kind of molecule is coming in close contact with each other. So what they did, so let us so you remember this picture I showed you, that you can imagine you have a D 483 somewhere, and D40, so maybe D 483 and R487. Maybe I will not make it because it's not relevant. So now, apart from D4883 and R, there are other interfaces which are connected here. So for example, glutamic acid and glutamine, and here also this two things are prominent: isoleucine 538 and isoleucine 499. So if you have one dimer and other dimer, that means another same dimer can form homodimer can form based on these interfaces. So what they did, they mutated few of this amino acid residues, and this were then only in one, only in one residue, or one molecule. For example, this is the right molecule where Q486 was changed into E. So this is Q2E, and then in the same molecule your I499 was changed into L. So basically they said it as a EL mutant. So because at 486 it is creating as a E, and then at 499 it is creating as L. Whereas in this case, in this dimer form, they are changing your glutamine 490 E to K, and then they are changing another isoleucine 538 to K. So this they mentioned as a KK mutant. So EL2 or EL or KK mutant. So that means from one side they are able to mutate this EL, and here they are able to make a KK. So based on this disruption, this is what they are making. So all the bonding which is taking place in between these two residues are disturbed. That means basically this bonding is based on the electrostatic and hydrophobic kind of interactions. So they are making these changes from glutamine 486 to glutamic acid, then glutamic acid 490 to lysine 490, and then isoleucine 499 to to leucine 499, and isoleucine 538 to lysine. So this kind of mutant they created, and they checked for the obligate nature of them, and they were able to check on the GFP kind of system where they integrate only GFP will be produced when there is a very much intricacy between these two molecules, when there is a cleavage in between these molecules. So what you can see, this is the heterodimer which is forming in normal cases. So you can consider it as 1 1 1 when both heterodimer or homodimers are formed. In this case, when you change only for 90 position, then you have reduced efficiency in heterodimer, but other here AA residues will not form because you have changed into this one. This will not form, homodimers will not form, but you can still find that homodimers of wild type will be formed. So that is why you have one residue. Now, in other case also, if you change Q486 to E, then this homodimer formation is reduced, in this case 0.49. Here this is reduced 4.2, and the heterodimer is up to 0.65. Now slowly they also mutated isoleucine 538, then the homodimer consideration is going down drastically, whereas the heterodimer formation is still intact. You do not have complete heterodimer. And when you introduce isoleucine 499 in this case also, then you have prominent only forming of heterodimer in this case, whereas you have very much reduced homodimers formation. So that means the problem of forming the homodimers are overcome by mutating the dimer interfaces which are present in these two. And same mutations are not done in same monomer unit, but different mutants. For example, this is KK and this is EL mutant. So basically these two residues were changed in both cases. And what you see here is the only heterodimerization prominent cases were found, either right and left or plus or minus or left and right. So these were found, but other homodimers were totally reduced in this case. Okay. So this was the first generation of your nucleases, where you have very efficient POC enzymes where only heterodimer is produced. And slowly later I found another enzyme, another paper where they also mutated one more residues, for example 496 in this case. So what they have shown here, this is the L mutant that I have shown already in this case. This is the EL mutation, right? So this is the EL mutation, and in place of original aspargine 496, in this case they have mutated aspargine into aspartic acid. So now this whole mutant is named as ELD. This one monomer contains these two and these three amino acid change. So here one monomer had EL mutant and another monomer had KK mutant. Whereas in this case, now after changing the aspargine to aspartic acid, you have ELD mutant, and in other case arginine 537. So this was initially histidine 537. And why they have changed it? Because if you see the structure, they are very much within the dimer interface and they are making prominent bonds here. So they have changed, instead of histidine, they have made KKR mutant or instead of R also they have made KK K mutant. So either R or either lysine. So here also they changes to I think arginine also. So they had tested two or three mutant. For in their case, aspartic acid were giving them prominent results. So they changed it to ELD mutant and KKK mutant or KKR mutant. So what they found after these checking, so all this, so initially this was from the earlier significance EL to KK changed. Now they checked ELD to KKR forming of heterodimers was highest in this case. And then ELD to KKK mutants also behaved properly, but it was less compared to KKR mutant. So basically, then they have found even the zinc finger activities was much better when these ELD to KKR mutant was there. So this was the second generation, and this is mostly what people use in the experimental system. And with this, they are overcoming the issue of your what you say the homodimerization formation, and now you have only heterodimers or obligate heterodimers are produced, and based on this you have very much reduced off targets effects. So with this, I am almost at the end of this Talon lecture with the fork enzyme. And what we have seen so far, that tail, or that means avirulent gene protein, binds to DNA in a novel way where only the box corresponds to its repeats that we have seen, and only the 13th amino acid defines the specificity. Importantly, the N-terminus sequences up to 127 amino acids are important. And for making obligate heterodimers, you can create this kind of mutation, and specifically this mutant work giving you highest efficiency ELD to KKR, and it reduces off targets. So now it is open for questions if you want to ask. With this, I'm completing the Talon chapter. Okay. Yes, Sam.
With respect to zinc finger, I mean, you mentioned it, Alan, though that they have an NLS. With respect to zinc finger, how exactly are they, like, if suppose if you are electroporating them, how exactly are they going inside the nucleus?
Okay. Zinc finger proteins, right? So zinc finger proteins, if you see, they are also transcription factors. So they will go into the nucleus. So mostly most of the transcription factors they work they go into the nucleus, and then they try to upregulate the genes, and we have already seen that these protein were isolated where from the nucleus, right, from the xenopus egg cells. So basically it transcribes your 5s gene. So you want to ask that what specific sequence are important for its nuclear domain?
Yes, ma'am. For localization purposes, like for example, we have an NLS in talents, but for zinc finger, we
Yes.
didn't really discuss something of that sort. So
Okay.
I was a little
So
confused.
we didn't discuss anything about NLS sequences, but I can find and come to your question there, because I currently I don't know what are the specific sequences that defines the nuclear localization, but we can I can check it.
Sure, ma'am. Ma'am, another question. So this is
Yes.
in reference with the repeat domains. You said that the 12th and the 13th. Initially, the thought was that 12th and 13th is what is taken care of in a repeat. Yeah, 12th and 13th amino acids, and eventually we found out that 13th is the one is which is base pairing. So if we mutate the 12th amino acid, does it really affect the specificity as such?
It will affect the structure actually. It is important for the structure because the 12th, if you see, always folds back. Whatever the repeats I have shown, so always all the 12th will fold back and stabilize the loop. So if you mutate the 12th one, it will affect the structure, and ultimately it will affect the spacing between your 13th position to the base pairing, and because of that you will lose the contact.
Okay, ma'am. Thank you, ma'am.
Any other question? So I think today you have another class from 4 o'clock, right? So tomorrow I will take a lengthy class. Maybe I'll go a little longer and we'll start with CRISPR chapter from tomorrow onwards. Okay. If there are no further questions, then I close this lecture today for today. See you on tomorrow. See you tomorrow then, same time. Thank you.